The overall goal of this assay is to determine the circadian period in Arabidopsis lines by measuring luminescence emitted from transfected protoplasts.
This method can help answer key questions in the field of circadian biology, such as which genes feed back into the timekeeping mechanism itself.
The main advantage of this technique is that it allows rapid analysis of the circadian period of any Arabidopsis mutant without the need to generate stable transgenic lines.
To begin this procedure, label a petri dish and add to it 10 milliliters of filter-sterilized enzyme solution. Fix 4 strips of autoclave tape with the sticky side up on a clean lab surface and mark the size of the petri dish on the strips. Collect plants from the growth room. Cut leaves of 4-week-old plants and press the upper epidermis onto the autoclave tape so the lower epidermal surface is facing up. Put strips of magic adhesive tape onto the lower epidermal surface. Using a 15 milliliter conical tube, gently press the strips onto the leaves, taking care not to crush the leaf tissue. Carefully pull the tape off to strip the lower epidermis and expose the mesophyll cells. Next, cut the autoclave tape to fit the petri dish and use tweezers to move the tape into the petri dish and float it on the enzyme solution with the leaves facing down. Rotate at 60 RPM on a platform shaker for 60 minutes, during which the protoplasts will be released into the solution. After 60 minutes, use tweezers to remove